Hi, and welcome to Mr. Fisher's Biology Podcasts. Our next podcast is on the biochemistry of cells. Just as an overview, if we look at organic molecules that we find on the planet, a couple interesting facts. Sugar is an organic molecule. Americans, and we can be lumped in there as Canadians, consume an average of about 140 pounds of sugar per person per year, which is a lot. The most abundant organic molecule are found in trees, and that is cellulose, and that's what we find in plant walls. If we look at this molecule right here, which is DNA, every body cell has about 2 meters of DNA. Now, for those people that want to do the math, there's about 70 trillion cells in a single human, so you can figure out how much DNA we actually have. These guys right here, they produce a ton of an organic molecule which we call methane, and right now engineers and scientists are looking at ways to make that a viable fuel source. So in order to talk about the biochemistry of cells, we need to first look at the molecule water. Because we're made mostly of water, and water is essential to most of the reactions in our body. Now, water has got some very useful properties. Water is a polar molecule, and so what that means is if I were to draw just a chemical shell of a water molecule, I would have a very big oxygen molecule, and then I would have two smaller hydrogens, and they sort of arrange themselves like that. Now, if you were to draw a mini equator across our water molecule, you'd find that these hydrogen atoms are a little bit more positive, and this oxygen is more negative. And so this creates poles, a positive and a negative pole. Now, why is that important? Well, if we look at the properties of water, the polarity allows for properties like cohesiveness, which is water's ability to stick to itself. It also allows for adhesiveness, which is water's ability to stick to other surfaces. So we already know that surface tension, which is a part of the cohesiveness of water, allows for some insects to walk on top of the water. We know that we see dew on plant leaves in the morning, and that's from the adhesiveness of water. And more importantly, or another important property, is transpiration. So plant transpiration allows water to conduct from the roots all the way up into the leaves, and that's using both cohesiveness and adhesiveness. So if we look at carbon-based molecules, we're really starting to talk about organic chemistry, and we're not going to go into organic chemistry in a great detail in a biology course. But most of living things are composed of carbon-based molecules. Now, if we quickly look at carbon, there's some useful properties about carbon that we can look at. First of all, it has 4 electrons in its outer shell that holds 8. So its valence is 4. Now, if I look at a carbon molecule and do a quick Lewis dot, you'll see that there are 4 electrons that are in the valence. Now, what does this mean? Well, this means that carbon has the ability to share electrons and form covalent bonds. Now, covalent bonds are very strong bonds. Remember, there's ionic bonds, and those are a little weaker. There's hydrogen bonds, and they're even weaker than that. So carbon has the ability to form these covalent bonds, and they can form different types. So carbon could form a single bond. We know that carbon forms double bonds, and carbon can even form triple bonds. And this allows for a variety of different molecules with different shapes, which we'll be looking at in just a little bit. So the simplest carbon compounds are known as hydrocarbons. They contain only carbons and hydrogens. Hydrocarbons are used in fuels. So gasoline is an example of a hydrocarbon, but also within us as a fuel source for us is fat, and fat is also a hydrocarbon. Both provide a ton of energy. So when we start to look at large molecules, we need to get some definitions down. Large molecules we call polymers. And polymers are built from smaller molecules called monomers. Now, biologists call polymers macromolecules, meaning big molecules. Okay, so these are big molecules that we're going to be looking at. In order to understand how we get big molecules, we link monomers together by a process called dehydration synthesis or condensation synthesis. Essentially, what we are doing is we're moving a molecule of water. So if we look at our two molecules right here, this guy and this guy, if we take an OH out of one and we remove a hydrogen on the other, we combine those together, they form our water molecule, H2O, and it creates a bond that exists between these two molecules. That's dehydration synthesis. The flip side is breaking them down. And breaking them down, that's hydrolysis, which is just the opposite of dehydration synthesis. So in dehydration synthesis, we are taking away water. With hydrolysis, we're adding water. So we're looking at a bond right here. If we add a molecule of water to this process, we break these two sugars into two. And our body does this in a process called digestion. So essentially, digestion is a whole bunch of hydrolysis reactions that break down larger molecules into something our body can then absorb. So there are four different categories of macromolecules that we find in cells. And we're going to look through the four in a little bit more detail. So carbohydrates is one of them, lipids, proteins, and then nucleic acids. So let's look at carbohydrates. So carbohydrates include things like sugars, which we find in soft drinks. They also include starch, which we find in things like pastas and potatoes. The monosaccharide is the monomer. Okay, so the monomer for carbohydrates is the monosaccharide, the simple sugar. Okay, monosaccharides typically have the same chemical formula, but they are different structurally. So there's three main monosaccharides, and those are glucose, fructose, and galactose. There are a couple others, though. This one, which we'll talk about later, and this one, which we'll also talk about later. Deoxyribose and ribose, which we find in DNA and RNA. But both of those are sugars. They're monosaccharides. So glucose we primarily find in sports drinks and energy drinks. Fructose is fruit sugar. It's contained in fruits. Honey, which is another type of sugar, has both glucose and fructose. And then galactose is milk sugar. Now, do not mistake that for lactose, and we'll talk about that just a little bit. But the way to remember sugars and the way to identify sugars is this. And make a note of this somewhere within your notes. It's a really easy way to remember. All sugars, for the most part, end in OSE. Okay, so it's an easy way for us to identify if something is a carbohydrate or if something is a sugar. Now, sugars do a very important thing, and they will form ring structures when they're in aqueous environments or watery environments. Okay, so glucose will form these ring structures, and you've probably seen these before, maybe if you've taken a chemistry course. Now, sugars themselves are fuel. They're fuel that our body uses for cellular work. And this important molecule here we will come back to in another podcast. ATP is produced from the breakdown of sugars. Okay, so the monosaccharide was our simple sugar, it was our monomer. When we link together monosaccharides and link them into twos, we have disaccharides. Disaccharides are double sugars. We join two monosaccharides together, and the process that we join them, remember, is condensation synthesis. Now, an important bond to just take note of, the bond that exists between carbohydrates is called a glycosidic bond. And there are some very common disaccharides, sucrose, which is table sugar, lactose is milk sugar, and maltose, which we find in grain sugars. So sucrose is a glucose molecule and a fructose molecule. Maltose is composed of two glucose, and lactose, remember we're going to come back to that, lactose is made of galactose and glucose. Now if you're lactose intolerant, it means that you don't have the enzymes necessary to break lactose into its monomers, and that's going to cause some digestive issues in your body. Now, when we string together many monosaccharides, we get polysaccharides, which we also call our complex carbohydrates. Examples of polysaccharides include things like starches, which we find in plants. Animals will store sugars in the form of glycogen, which is a polysaccharide. And plants also use another polysaccharide called cellulose to give their cell walls some strength. So plants will store excess sugars in the form of starch, and starch is a polysaccharide. And we find this in potatoes and grains, which is major sources of starch for the human diet. Glycogen, again, is how animals store excess sugar. Both glycogen and starch are actually very similar in structure. They're both made of strings of glucose monomers added together. Remember that cellulose was the most abundant organic compound. It's a major component of wood, which is very important, as we know. Also, it's known as dietary fiber. So fiber that we need daily comes from plants in the form of cellulose. And our body can't break down cellulose, which is why it is a fiber source. Simple sugars and double sugars dissolve readily in water. This is known as a hydrophilic or water-loving molecule. Okay. Now the flip side to those are hydrophobic. Lipids do not mix with water, and so they are hydrophobic. Okay, and so this is a fat molecule, and we all know that when we add fat to water, they don't mix. So let's look at lipids. Lipids store energy. They help to insulate body. They also help to cushion and protect organs. There are some different types of fatty acids. Now, one thing to note is that there's really no monomer to note within the lipids. There's just different types of lipids. So we have saturated fatty acids. Saturated fatty acids have maximum numbers of hydrogen bonds, and unsaturated fatty acids, they don't. And so they have a double bond between carbons. Now, what does this do? Well, saturated fatty acids would look something like this, and unsaturated fatty acids have this double bond, so they have a kink. Now, if we look at types, most animal fats are very high in saturated fats. They exist as solids at room temperature. Okay, so things like butter and shortening, so on. Oils, plant oils, tend to be very low in saturated fatty acids. They exist as liquids. So they're unsaturated fats, and they would be liquids at room temperature. Dietary fat is the molecule known as a triglyceride. Triglycerides have a glycerol, which is this right here, and 3 fatty acid chains. And there's 1 fatty acid chain shown, but there would be 3 of those attached to this triglyceride. Now, another important type of lipid is the phospholipid. Phospholipids are made of 2 parts. One part that is hydrophilic, which is this nice polar head here, and then we have 2 tails that are hydrophobic. Now, phospholipids are a very, very important part of a cell membrane. And a cell membrane is composed of a bunch of phospholipids that arrange themselves like so. This creates, remember from your grade 11 biology, this will create sort of a double membrane, okay, which makes your cells selectively permeable. Another type of lipid are the steroids. These are these are bent to form fused rings. The most important steroid, or the base steroid in our body, is cholesterol. And cholesterol is, or helps to produce other steroids, which are very important, like estrogen and testosterone. Let's look at proteins now. Proteins are made of monomers called amino acids. So the amino acid is a monomer in proteins. And there's actually 20 different amino acids, and they're all linked together in different orders, and the bond that exists between them is the peptide bond. Now, the purpose of proteins, they are used as structural to help build cells. There'll be hormones, there'll be enzymes, and we'll have a separate set of notes talking about enzymes, and they basically do most of the work in the cell. Now, some other important proteins, we know that insulin is a protein, helps to regulate blood sugar. The cell membrane contains tons of proteins. It contains proteins that are on the surface and proteins that are all the way through. We have receptor proteins, which will help cells recognize each other. So proteins are a very important part of a living organism in the cell. Okay, our last macromolecule is the nucleic acid. And nucleic acids, they store hereditary information. So they contain the information for making proteins. And there's really just 2 types of nucleic acids. There's DNA and there's RNA. Okay, so a nucleic acid, it's made up of monomers called nucleotides. Okay, so the monomer for the nucleic acid is the nucleotide, and this is a nucleotide right here. Okay, nucleotides have 3 main characteristics. They have a phosphate group, which I'll circle in red. They have a 5-carbon sugar. Okay, so this is deoxyribose, so this would be a DNA nucleotide. And then they have a nitrogen base. Now, the nitrogen bases can be, in DNA, they would be one of these 4, adenine, guanine, thymine, and cytosine. And we'll come back to this in the next unit. We'll do a lot more with that. So nucleotide monomers are joined together by the sugars and phosphates to form this backbone. And we can see the bases kind of out to the side here. On a DNA molecule, those will actually form, 2 strands will form together to form what's called a double helix. Now, ribonucleic acid is the other nucleic acid. The big difference is it's got ribose sugars, and instead of thymines, RNA has uracil. Okay, so a nitrogen base is different, as well as a ribose sugar. So the next 2 slides are a summary of key concepts, and they're actually very useful to add as a summary into your notes. So pause the podcast as we go through these, and or as you go through these, and create this table. And this table will be a useful study tool when it comes to understanding the macromolecules. Okay, and that's our podcast on the biochemistry of cells, and hope it was helpful.